third part of what is needed for to do an in vitro assessment of fish feeds, which refers to the in vitro digestion itself. Obviously, you cannot feed cells with pelleted fish feeds, so we need to do something with them before. And to tell us more about that, we will have Dr. Amir Bitan from Israel Oceanographic and Limnological Research. You're a researcher at that institution within the field of fish nutrition. His research interests include the development of genetic and in vitro tools for nutrition research, examining meats to increase nutrient utilization in fish, and studying the maturation of feeding behavior, appetite, and satiety. So please, Amir, the floor is yours.
Thank you, Trond. Can you hear me? Okay, so first, thank you very much for the university, for NMBU, for hosting this event for us, for the Fish AI. Thank you very much for that. So my name is Amir Bitan. I come from Israel Oceanographic and Limnological Research Center in Eilat, in Israel. And I would like to first present the team that worked on this project on behalf of IRLR. So we have Dr. Marcelo Chacon, who is also here. He has done most of the work that you will see in the slides, in the upcoming slide. Dr. Liat Koch, Professor Amos Tendler, and Bill Coven, who all participated together with us. So our task was to design an in vitro digestion procedure of fish feeds, but in the scope of fish AI applications. So what does it mean? Basically, it means a method of how to feed the fish AI, because we cannot feed the cells with regular pelleted feed. The cells, as Nicole just demonstrated, they come from the proximal intestine and the distal intestine of rainbow trout. So in those areas, the cells in those areas would be exposed to already partially digested feed and not a complete feed. So we wanted a method to feed those cells with the test diet. So since this is an AI project, so we decided to ask the AI how to feed the fish AI. And this is the AI idea of how this can be done. So basically, we want to in vitro digest fish diets, but we want the digestion of the diet to be targeted toward the cell culture applications. What does it mean? It means that we want to determine how to introduce fish feed to rainbow trout epithelial cells in a way that will best resemble their exposure to digesta in the fish intestine. So we do this by two sequential digestion stages, which represent the gastric and pancreatic digestion. And we ask ourselves, since those cells are coming from the intestinal area, how should the digesta look like when they reach those stages? So in analysis that we performed, we actually analyzed the metabolomic profile of the digesta that is found in different sections of the digestive tract of rainbow trout that were fed fish meal reference diet. And here in the graph that you can see here in the bottom, you can actually see the free amino acid distribution in vivo that were found in the feed. That would be the blue bars. And then in the stomach, you see that there is not much change between those two segments of the digestive tract, as you would expect. And then when we reach the proximal intestine, you can see higher amounts of free amino acids. And then as the digesta continues to pass through the digestive system, we can see that the levels that we can detect in the digesta are actually decreasing. So for us, that meant that we have to aim toward an intermediate digestion of the fish feed versus a complete digestion that you would expect to see. So if I will refer to what we just heard in the beginning of the day from Ramon. So the apparent digestibility coefficients that Ramon was describing us, those were analyzed from the feces of the fish. So that would mean that the analysis was performed at the end of the digestion. And here we are actually aiming to produce a digesta that will mimic more the more proximal parts of the digestive system. Another crucial point that we have to take in mind is that we are doing this in vitro digestion mainly to be able to expose the cells to the digesta. So that means that we are not going to remove any products of the digestion that is occurring in our in vitro digestion procedure, because we want the cells to be exposed to those nutrients, and we want the cells to be the ones that continues the digestion and also the absorption of those nutrients. And we have to bear in mind that the fish AI RT cells are actually out of their physiological context. So that would mean that we do not have any mucus layer that provides buffering microenvironments for the cells. And also we don't have the microbiome in our system. In that sense, we have to bear in mind that the high VD that we are producing, the in vitro digested feed, it must be safe for the cells. So that is the main purpose of us producing this digester. So in order to digest feed, we were using crude enzyme extracts. We prepared them from rainbow trout, stomach, intestine, and pyloric theca. And we verified the recovery of pepsin, trypsin, and lipase activities in those extracts. We were using these activity measurements also in order to normalize the amount of enzymes that we are adding into our in vitro digestion procedure. And we normalized it based on the activity units of those enzymes. So in that sense, we can have crude enzyme extracts coming from different fish that were fed the same diet during different trials, but we would like to normalize the activity level of those enzymes so we can have a similar production of digesta. At the end of the process, since we are exposing the cells to this digesta, we added some additional stages, which include enzyme inactivation. As I remind you, we don't have mucus on the cells. And also another step of filtration in order to create digesta that will be sterilized and can be used further for cell culture. We have tested during the project several conditions of in vitro digestion procedures. Here you can see an example of metabolome analysis of digesta that was produced by two IVD conditions. One, which is low enzyme substrate ratio and longer incubation time. The second is the opposite. And you can actually see that we get different end product by changing the conditions that we are using in order to do the in vitro digestion. This specific experiment also had two additional groups where we used actually commercial enzymes. So this will be pig enzymes. You will see them in the middle over here. Can you see my, sorry. The pointer. Thank you. So you can see them here in the middle. So you can see that they have distinguished metabolome profile, unlike when we use enzymes that are coming from crude enzyme extract from rainbow trout. So after we decided on the conditions that we want to proceed with in order to produce the in vitro digestion of rainbow trout feed, we continued and formulated 7 diets with assumed contrast effect of protein on a protein value. And we used several protein ingredients that are actually relevant to the industry, to the fish feed industry. So we were using fish meal as in the reference diet that we chose. We were using soya in several concentration. We're using feather meal, which is known to have lower digestibility, and we were also using gur meal. And we in vitro digested those feed using the fish AI IVD protocol and analyzed the outcome of it. So the first thing that we looked at protein digestibility, and this was done in a collaboration from NMBU with Nufima. And here we actually analyze by using size exclusion chromatography, SAC, the in vitro digested feed. And we ranked the different feeds by protein digestibility, where we were choosing to use the cutoff of 10 amino acid residues as the bioavailable fraction for the fish. And when we compared, when we look at the protein digestibility that was generated by our fish AI IVD procedure, so you can actually see that the reference, you can actually see the reference diet over here, which is a diet that is enriched with fish meal. You can see the gur high diet, the soy high diet, which has a very good digestibility. And you can see that the feather including diets, they have they are the diets with the lowest digestibility from all. When we compare this, so we use the same feed in order to perform an in vivo feeding trial, and following the trial, feces were collected, and the digestibility of protein and total amino acids were calculated, as Ramon explained in the morning. And you can actually see that between the model and the fish, we can see very nice correlation of digestibility. So you can see the better performing diets in terms of protein digestibility. And you can see that the feather diets are expressing lower digestibility in both the fish and also the fish AI IVD. But when we look at the actual quantification of digestibility, we can see that the actual numbers of digestibility that we see in fish are higher than ones that we see in the fish AI IVD. And you can see that the correlation between the two, between the in vivo experiment in rainbow trout and the in vitro digestion of the fish AI, was quite good. We also tested how our model, the in vitro digestion model for the fish AI, how it stands in correlation with other known models for protein digestibility. So another in vivo model that we tested was the mink model, and we can see very good correlation between the in vivo mink model and the fish AI IVD. But also we can see a nice correlation between the fish AI IVD model and the InfoGest, which is a mammalian in vitro digestion model. So we exposed the diets that were in vitro digested by our procedure to cells, and we found out that the cells cannot tolerate this digesta, and that the digesta has negative effects on the cells. So following some further analysis, we figured out that the problem is the addition of bile salts. This is one of the things that the cells cannot tolerate. So we had to remove this additional step from our in vitro digestion procedure, because as I remind you all, the purpose was to produce digesta that is also safe for cell culture applications. So this is a burden that we had to take because our main goal was to test those diets on the cells. So when we do this, and we analyze the digestibility profile of the diet when we are not using bile salts in our in vitro digestion model. So we can see that we have an effect on the total digestibility that we can observe. So the digestibility level goes down. But we see that the overall ranking remains the same. So this is the trade-off that we have to pay in order to be able to expose the cells to our in vitro digestion diets. You can see that the correlation between the fish IVD and the trout in vivo experiments. You can see the correlation is not as good as the in vitro digestion with bile, but still this is what we ended up with. Another method to rank the diets based on the digestibility, because we are thinking also about the end users of this platform, and not everyone, not every lab or not every feed company will have access to performing this SEC analysis in order to study digestibility. So we tested several on-the-bench methods that can assist in estimating the protein digestibility of the diets. And eventually we selected between all the analyses that we performed, we selected the OPA method. So OPA is interacting with primary amino groups to create, in the presence of thiol, to create OPA amino acid complex, which can be quantified. And we can see here that this analysis is quite rapid, and it's also sensitive to different conditions that we are using. And here you see the same two conditions that I elaborated earlier on. So one is high enzyme substrate ratio and longer incubating time, and the second is the opposite. And you can actually see that we have a nice differentiation between those two conditions using these very easy methods of detection. So later on, you will hear about another in vivo trial using 5 contrasting diets, including fish meal, soybean meal, feather meal, and 2 other components coming from single-cell proteins. But what I wanted to show you is that that also when we are testing different diets, diet ingredients, we can also get different ranking, different protein ranking or digestibility ranking using also this OPA method. And I can already say in advance that this ranking that was obtained here was the same as the one obtained in vivo with rainbow trout fish fed the same diets. But you will hear about this more later. Since our purpose is to expose cells to the digesta, so the next step was to take the in vitro digested feed and expose rainbow trout cells to it. So we have 2 types of cell lines: the rainbow trout, the proximal intestine cell lines, and the distal intestinal cell line. There are several analyses that can be performed in order for us to understand the effect of the digesta on the cells. So over here, you can see some examples. You can see examples of first how the cells look like when they are exposed to different digesta concentrations. And you can see the upper panel is the before and the lower panel is the after. This is the RTPI cells coming from the proximal intestine. And the first thing that you can see is that there is a concentration effect from the diet. This is the reference diet, so it's a regular fish meal diet. So you can see that when we expose the cells to the digesta as it is, the cells cannot tolerate it. Okay, so that means that we have to dilute the digesta in order for us to be able to perform in vitro assays on the cells. We have an hypothesis that this has to do with the osmolality level of the different digesta that is being produced. Also, we can check using some known cytotoxicity methods, what is the effect on different things in the cell. So we have here this MTT assay, Almar Blue assay, CFDA assay, and natural red, which can give us some information about the metabolic status of the cells, the intact of their cell membranes and the lysosomes, etc. And by that, we can actually see the same responses we are seeing here in the micrograph below. We can see that there is a digesta concentration effect on the status of the cells. And we have done this also in the distal intestinal line, and the take-home message from these slides is that we can do this analysis with different digesta, but that we have to take in mind, we have to consider the digesta concentration when we are doing dose exposure experiments. So this is part of calibration that we'll have to go through when we expose the cells to different diets. One of the main purposes of the fish AI is to study the health effects or the response of the intestine to the different diets. So one of the methods that we have routinely used is to evaluate the cellular integrity, because the intestine integrity is an important parameter that we want to keep, and we want to keep track of in vivo and in vitro. So in order to do this in vitro, we were using a procedure which is called transepithelial electrical resistance, or in short, TEER. And in this type of analysis, we have an electrode that is put on the transwell. You will have the cell layer, and if the cell layer creates a functional barrier, you will have the resistance will be higher between the upper and the lower area of the chamber, because fluids cannot pass through because of this barrier. And you can see here several examples how different concentration of in vitro digested fish meal, fish meal based diet, the reference diet. You can see the before and after, and you can see that when you are using the reference diet, there is no considerable effect on the electrical resistance of the cellular integrity. Another way to measure this is by using molecules that are not supposed to pass through the barrier in a very easy way. One of them is Lucifer yellow. So this is another way to evaluate the cellular integrity in our system. When we are looking at actual diets, and here you can see 4 conditions. So L15 is the medium, the regular medium that the cell grow in. You will have fish meal, based diet, soybean meal based diet, and feather meal based diet. These are the colors. And you can see that when you analyze the T response after exposure to in vitro digested feed, you can see clearly an effect of the different diet, which is also digesta concentration dependent, as you can see here. Another way to look at the response of the cell layer to the in vitro digested feed is by analyzing the cell coverage inside the transwell, as you can see here in those images, comparing cells that are exposed to L15, which is the cell medium, fish meal based diet, and soybean meal based diet. And you can also, we can also look at the morphological analysis of this cell layer, and we can actually see the differentiation that is occurring because of due to exposure to different diets. We can also test the permeability of the cell layer. And by that, we can use, for example, FITS-dextran, so in different molecular sizes, either 4 kilodalton or 10 kilodaltons. And here you can see the same diets. We now have an additional control group, which means an insert that is devoid of cells, so fluids can go freely through it. And we can actually see that as we go up in concentration, we can actually see that we have a different response of the cell layer to those diets that corresponds also to the tier that we are seeing here. We also can evaluate the permeability to dipeptides. For example, these are the main nutrients that are absorbed in the intestine. And here we are using the beta-allylisine AMCA dipeptide. So it's basically a peptide that is fluorescently labeled, and we can actually quantify the amount of beta-allyl AMCA that is in the cells and also that goes through the basal to the basal part of the transwell. We can also look at fatty acids using the body PE molecules. So here, and those molecules will come in different lengths. And by that, we can also track the permeability to fatty acids of the different diets. So to summarize what I just described, so we describe a procedure to introduce fish feed to rainbow trout epithelial cells. The IVD was performed by 2 sequential digestion stages that represent the gastric and pancreatic digestion. We aimed for intermediate digestion and not complete. Without digestion product removal, that will be performed by the cells, with no significant cytotoxic effect, and without altering the integrity of the cell monolayer, at least for the reference diet at certain concentrations. So that will be the base level, and from there, we can compare to other diets. We also defined, I didn't show it here in the presentation, but you have to know that we also defined a procedure to lyophilize and reconstitute the IVD feed. That means that we can ship it more easily, store it, and use it later for with our cell cultures. We produce diets with assumed contrast effect on protein value. The in vitro protein digestibility profiles of these diets show high correlation with the in vivo digestibility in rainbow trout. And we demonstrated method to study the effect of diets in the fish AI following their in vitro digestion on cell viability, metabolic activity, cellular morphology and integrity, and also some aspects of intestine functionality. And all of this will be presented later on after the lunch break in a more elaborated way on a big feeding experiment that we have produced. Thus, we demonstrated the applicability to test the effects of novel feed ingredients on the cellular response and on the intestinal barrier function. And with that, I would like to end by thanking all the consortium members that contributed to this work, and also thank NMBU collaborators from NOFIMA for helping with the second analysis, and also Francisco Moyano for giving us useful advice during the early stages of the in vitro digestion procedure. Thank you.
Thank you very much, Shamir. We will soon have a coffee break. We have had some questions online, so I think I will take one of them at least for you, or maybe some colleagues, but we can also bring them up later.
The others you can email me.
Yeah. Oh, we have time to talk afterwards. Let me see, maybe I can read it over here. Okay, so one question from the online attendees relates to the use of bile salts, and it's referred to another study that is published by us actually, or by my colleague Guro here, independent of this fish AI work, where it was shown that exposing similar type of rainbow trout gut cells to bile salts, either obtained directly from the gallbladder or pure taurocolate, the cells retain normal cell morphology and viability. And the question is, is wondering what you think went differently in your studies that show that you couldn't use the bile salts on the cells?
I'm not sure.
But perhaps Guro can answer to that, because this has been a collaboration.
Okay. So the main difference is the concentration. The concentration that we needed to add for the in vitro digestion for it to have an effect was much higher. While also in this paper, we showed that high concentrations of the bile salt were toxic to the cells. So the concentration that we ended up in the paper saying that the cells tolerated, we actually tested that for the in vitro digestion to add it, but we didn't see any difference between adding that low concentration and without the bile salt. So when to simplify the procedure, we ended up using without the bile salt, because we could not have the initial high concentration that's in the InfoJust model. Okay?
Thank you. Okay.